Hello Indrani, how are you? My voice is audible to you. Just let me know if you are able to listen to my voice. Is it audible to you, Indrani? Yes, I am fine. I am fine. How are you? Yes. So my voice is audible to you or not? Yes, I received your abstract last day. No issues. Yes. Okay. Is there any type of noise in between my voice or my voice is audible? My voice is audible to you? So my voice is audible to all of you? Please let me know my voice is audible. Okay, okay, thank you, thank you. So how are you all? How are you all? I hoping everyone will be fine. So please let me know everything is fine. Just how are you all? Everything is fine on your side? My voice is audible to all of you? Okay, okay, good, 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 good. Definitely you have 1 or 2 days along with you, so you must have done some part of your review paper. I think so. Yes, I am fine. Aniket, thank you. Yes, yes. Good evening, Valina. Nischal is there. Somnia is there. Yes, I am fine. It's just I am feeling a little bit cold today. Otherwise, I am good. So just want to know how are you all and whether you have started writing the topic, whether you start writing the review paper, have you selected something or not, then please let me know. And sorry about the my video because I'm on some another room in which there is not a proper light, so maybe uh, you are getting me a little bit more with full of light. It is because there is only one lamp here and rest the room is dark. So but there is a good signal, that is why I came here. So just let me know everything is fine on your side. Yes, Nikita, good evening. Ankita, very good evening to you. Yes, Aniket, I reply to Aniket. Yes, Deepti is here, Aradhana is here. So I think that all of you must have start writing a little bit about your review paper, or at least you have selected your topic. So if it is like that, then please let me know, have you selected your topic or not. If it is selected, then are you able to write the abstract on it or not. So please just let me know so that other students will also join with us. At that time we are just talking with each other. Okay, okay. Indrani is saying your voice is audible. Okay, thank you Indrani, thank you a lot. Okay, okay, very good Valena. If you have started writing a little bit on your topic, it's very good. Yes, good evening Pooja. Yes, Arif, it is little bit good today. Actually, I have a fever and I am feeling little bit cold. Otherwise, it's okay. Yes, 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 I am fine, Megha. Yes, please, please, all of you, teach me, and all of you, all the other students, please start writing a little bit about the paper. Even if you have if you first of all you have to select the topic. So if you are able to select the topic, then the rest things are very easy. So selection of a topic is very important. So just select your topic and then search the papers which are related to your topics, and then you have to take the content from only those topics. Okay, so you have to take the content from the topics, only from those topics. Yes, very good Arif Qureshi. It's very good that you have selected your topic and now you are just searching the literature, because searching the literature is the most important thing which we actually forget. We always want to do it fast, but searching the literature and taking the good content from the literature is the most important thing. Yes, yes, Shrinesh. Okay, okay. So Shrinesh has started writing the abstract. Very good, very good. Then we have Nikita. Nikita is saying which topic we can take. Nikita, it's your own wish on which topic you want to do the work. It's your own wish. I am not forcing you. You can select any topic of your point of interest. So you can take any topic. Yes, Indrani, I will mail you personally. I will mail you if I am finding it suitable. Then I will ask you to proceed it. If I want some, I want some my suggestions. Then I will definitely tell you. But I will mail everything to your you. I will reply to your mail basically. No, 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 Vanina, it's your own wish. I already told you on the very first day that you are quite mature enough student, so you can take your own topic also. But some students want to that I suggest them. That is why I put it in the group. Otherwise, you have your own choice to select your own topic. No issues at all. Yes, yes. Yash Raj is unable to select the topic and he wants my help. But Yash Raj, actually, if it will be so nice of you if you just introduce yourself, like in which class you are and which subjects you are pursuing. Then I will help you whether you are in zoology or in biotechnology, and then I will help you in selecting the topic related to your own field. Okay. So if you are able to tell me that what is your background basically, your class or your subjects, then I will help you. No, no, Mega, actually, if you are able to select the topic, if you are able to, you have select your topic, then you have to find the literature related to that particular topic. So if there is a topic for molecular markers, okay, so you have to at least select 10 to 15 literature or 10 to 15 review papers on the molecular markers, and from all these papers you will get idea that which content or which thing you want to write in your own paper. So that is why I am giving you the time for abstract, that is 2 days or 3 days, in which you have to search the whole literature. Okay, so by searching the literature you will be able to get the good content basically. So much light is there in my eyes. I am not able to read your comments even. Yes, yes, there is a deadline basically, like today is 2nd of November, and you have to submit it by Friday basically, by 6th of November or within this weekend, so that even you have to send it me before, because you have to submit it by 10 or 11th of November, so it will be better if you send the abstract within this week. Yes, yes, Bhumi, please, it will be nice of you if you are able to share your queries also here. I am just reading your messages, and I will definitely reply, no issues at all. Radhu is there. Radhu, good evening, how are you? Radhu, your name is very unique and it's very adorable also, Radhu. Very nice name. Yes, Yashraj is pursuing BSc Biochemistry. Okay. And in biochemistry, what is your point of interest? Yes, like you are pursuing the biochemistry, and what is your point of interest here? Nikita, yes, Nikita, if you will be able to tell me that from which class you are, or you are pursuing in which topic, so then I will help you in selecting some topics also. Megha is saying, okay ma'am, got it. Okay, okay. Ayushi is here along with us. So Ayushi, good evening. And Amrita is saying, yes, yes, Namrita, please share your queries here also. Rohan is here. Like Rohan, you are from B Pharma, you are pursuing pharmacy, so you can take any drug, like you can take medicinal plants as a drug, some type of this type of thing, a review that you just tell us some medicinal plants which are used as a drug, or which are basically used to cure some diseases like maybe diabetes, maybe any type of allergy or any type of, you know, even for cancer also. So if you are able to do such type of review paper in which we are able to know the medicinal plants, or you can say any type of uh, drugs which are there, which are not the basically chemicals, but they are very natural in their content. So it will be very nice of you if you are able to write the review on these things. So you can take medicinal plants as a drug, that how they are helpful as a drug, or how they are helpful for curing the diseases in humans, as well as in case of livestock also. So yes, Sadhu, I am fine now. Just wait for 1 minute. I am not able to read it. Okay. I think it's okay now. So sorry for the lightning today. So yes, Bhumi is saying. So Rohan, I already answer your query that kindly pursue something on medicinal plants as drug. Then Bhumi is saying, ma'am, can I take bioplastics? Plastics? Yes, definitely, Bhumi. Please take bioplastics as your topic. No issues at all. Yes. Please, Indrani. It will be so nice of you if you are able to help us in writing this review process. It will be so nice of you. Sharja is saying good evening, Sharja. How are you? Yes, Shahad. Just please be in contact with Indrani. She is pursuing her PhD, so she will definitely help you. Pooja is saying, ma'am, when was the submission date of abstract? So Pooja, you have to submit it within this week, basically up to Friday. You have to submit it. That is 6th of November. Then we have Shrinesh along with us. No, there is no problem of taking two students taking up same topic unknowingly. Even if you have your friend or you want to take it with someone, then you are free to take, you are free to make a group of two. No issues at all. Yes, Sharja, fine, ma'am. I am fine, dear. I am fine. Indrani is saying I am there, always helping for youngsters. Okay, Indrani, that's so nice of you. So then we have Shivangi. How are you, Shivangi? I am good, thank you. Yes, Mayuri, please select the topic of CRISPR. This is a very hot topic, or it's a very it's the latest topic which is now in the news. So please continue with it if you are able to. We have Aryan along with us. So Aryan. So Aryan, how are you? How are you, Aryan? Yes, Tanvi, good evening to you, Tanvi. How are you, Tanvi? I hope you all will be fine. So just waiting for 1 or 2 minutes more so that others will also join with us. Then we will start our session. Yes, Ishani, because when you will send me the abstract, but before sending me the abstract, you have to read the whole content or whole the literature, basically 10 to 12 review papers. So you have done the searching part within this week. Okay. Then after this week, that is after the 6th of November, that is in the next week, you will be able to write the whole topic. It will not be difficult for you because you already searched the whole content. Okay, so please don't worry. Yes, Rohan, you can take anything, but basically I'm suggesting you any drugs basically, so you can also use the synthetic drugs or something. No issues at all. Yes. Why are you asking if I am a ma'am allow? I am always helping. I am always there for your help. So if she is also pursuing PhD and she is willing to help you, then please take her help. She will definitely guide you the best. No issues at all. So please just be in touch with Indrani ma'am, or she's your ma'am basically. So just be in touch with her and just get the knowledge. No issues at all. Thank you, Indrani. Thank you. Who is this living a normal life? Who is this? What's your good name? Living a normal life. Yes, you can pursue it on enzymes, no issues at all. But what's your good name? Living a normal life. Is it your channel? Aryan is good. Yes, Mumi. Please, if you have any doubt, you can share with Indrani. No issues at all. Maximum and minimum pages, at least you have 8 to 10 pages at least. Yes, Pratik. Actually, I'm just waiting for few more students to join. Then we will start the session. Just 1 or 2 minutes more, please. Then we will start there. No issues at all. You are thinking of gut microbes and depression. Yeah, it's very unique topic. I also want to know what basically this is. So if you have some knowledge, then please kindly pursue it, and I will definitely check everything and we will send it to the journal. No issues at all. Yes, Shivangi, I know that some of you have to submit your 10-day lecture report to your teachers. So I will definitely send every PPT, everything which I have to you. No issues at all. But just let me complete the PPTs, then I will share it. Yes, living a normal life is Anu. So all the best, Anu, for your channel. And according to its name, it's maybe something about positive thing or positive vibes in your life. Maybe it is like that. So I'm just wishing you all the best for your channel. And I just please send me your this link. I will definitely watch your channel. Okay, send me your link. Oh, Stuti is saying, ma'am, when will the session begin? Just okay, dear. I'm just opening the PPT. Okay. Actually, I'm not able to get to see the laptop even because so much light is there on my eyes. Okay. So today we have to start the topic which is biological databases, and let me show you one thing also. Like this is the introduction of bioinformatics which we have done in our very first class. Then we have biological databases which we have to start today and which we will do today. Then we have to do bioinformatics database files format. Then we have sequence retrieval from protein and nucleic acid databases. Then we have sequence search by FASTA and BLAST. Then we have pairwise alignment. We have multiple sequence. Actually, all these topics, they are, you can say, they are interconnected with each other. So there is not even a single topic. So all these topics are basically, they are in, you can say, they are interconnected. They have the connection with each other. So within these 1 or 2 days, I will try to cover all these topics. Then we will move to our final topic, which is the mega software. So we will move to our final software after completing these small topics, because this is the general knowledge to you about the platform, about the bioinformatics basically, but our main focus is on the mega software. So I just want to dedicate 1 or 2 lectures specifically on the Mega software, because I want that you become master at least in Mega software. So in these topics I will cover in a little bit fast, or even I will take extra class also, because my main focus will be on the Mega 6 software. So that is why I also told you before that please I also told you before that please download the software and something. So I hope all of you have downloaded it, but I will start it this Mega 6 software after the finishing of upper small topics. Okay, so I hope you all will getting my point. Yes, I'm just going to start the session. So Indrani, students are asking for your number, so please convey your number to all of them so that they will get some knowledge or something from you. I hope they will definitely get something, and they are able to, you can say they are able to write the good review paper. Okay, so I think lots of students are here now, so I am just going to start the session. Then we will take the break for some minutes, and then we will discuss again if you have any other query related to your review paper. Okay, so now we are starting our first topic. So is it clear to all of you? And then we will do the small topics first, like biological database or the sequence retrieval and fast and blast sequences. But our main focus is on Mega 6, which we will do in the last days, or we will dedicate our whole session on the software basically. Yes, I don't know how to adjust the screen. Actually, it is not the screen, it is the lamp right here, because the whole room is black. That means there is no light there. So that is why I am feeling so awkward today. How to I don't know. Let's hope this will help. Yes, Rajita Roy is confused about the topic, but Rajita, we have discussed everything within this 15 or 17 minutes before. So we will discuss it again personally also, no issues. No, let me start the topic now, because we are already very late. So Rajita, you have to tell me your class, in which you are pursuing, or your subject basically. Then I will help you regarding the selection of the topic. So Aishwarya, you all will discuss everything with Indrani at any time. So now we will start our session. Okay. So let me start it. Yes. So I am starting it from very basic, so that all of you will be able to understand it. I am just starting it from very basic things. So we have to do major databases in bioinformatics. What is a database, and everything we have to discuss. So okay, so what is a database? Basically, database are convenient system to properly store, search and retrieve any type of data. So in case of database, if you have the database, what's the function of this database? That it is convenient system to properly store, search and retrieve any type of data. So in the database system, we are able to store, and then we are able to search anything and retrieve any type of data. A database helps to easily handle and share large amount of data and supports large scale analysis by easy access and data updating. So we have this, we already discussed in our previous lecture also, that in the database we have large amount of data which is stored there. So we will put our, you can say, this data is easily accessible to all of you, and if we want to do something, if we want to add something in it, we can do it, and we are able to do the data updating. So we have the system for storage, for searching, and then for retrieving any type of data. So database helps to easily handle and share large amount of data and support large scale analysis by easy access and data updating. So we have to study about what is basically biological database. What is basically biological database? So biological database are libraries of life sciences information collected from scientific experiments. So basically it's not a journal data. We have do a lot of research and then we are able to get the data. Published literature, high throughput experiment technology and computational analysis. So it is that the data will be stored only if it is collected from the scientific experiments, published literature, high throughput experiment technology and computational analysis. They contain information from genomics, proteomics, microarray gene expression. So they contain the information at the gene level, at the protein level and also by using the microarray gene expression. So information contained in biological databases include gene function, structure, localization, biological sequences and structure. So that is why bioinformatics is such a vast field that we have so much of things to discuss. Like we have gene functions, we are able to understand about the functioning of the gene, we are able to understand the structure or even the localization of a particular gene, and then we are able to understand the biological sequences and their structure. So we are able to do the you can say by using the biological sequences or by using the structures we are able to identify anything, okay, or any organism or something is like that. So biological database or you can say bioinformatics is a vast field in which we are able to collect the data, and data is in the form of biological database which includes gene function, structure, localization and of course biological sequences and structures which we are able to study. So this is the biological database. So we have the information system, like we have some platform where we are able to search our thing, like we have Google, we have GenBank, we have so many of sources. So by we on these sources we will able to get the information, and then we have some query system when we want some type of query, and then data will be stored like in the GenBank. Like in the GenBank the data is stored. So we will be able to get the data, just by typing the title of a book, or even we are able to get the data by just by just putting the session number, by just putting the name of the species, you will be getting the data. So this is the thing that we have the information system which is your Google or some app. Then we have query system is also there. Then we have the data which will be available to us by just typing the you can say title of a book, or by typing the GenBank session number basically. So this is the thing that a sequence retrieving and manipulation network. We have databases. Databases have the retrieval system. So we are able to get the information in the form of sequences or in the form of any type of image. Then these sequences, these data which we get, we will put it on a software, like we will put it on a maker software, and from these softwares we are able to get the result. Okay, so how we are able to get the databases? We will get it from the GenBank, or if it is DNA we will get it from NCBI GenBank, from DDBJ and from EVI AMBL. These all are the platforms basically from where we are able to get the data. Or in case of protein, if you are working on a protein, if you want to get something like 3D structure of protein or something is like that, then we have SwissSpot, we have PIR. These are the platforms on which we are able to get our data, and from this platform or from these sources we have to take our only the information which we want. So we have to take only that information, like this is the information which we get in the form of sequence, in the form of image. Then we have to check it in the software. So today we are discussing about only this thing: databases, retrieval system and information. So when we are able to get the information, then this information we have to put it on a particular software, and from this software we are going to able, or we are able to get the results. So I hope this whole scenario of bioinformatics is clear to you, that from the database we are able to get the sequences, or you can say information. This information will be put on some software, and from the software we are able to get the results. Okay. And of course there are some format of putting the sequence. There is some pattern basically of how to put the sequence. The sequences can be put in a specific pattern by which the softwares are able to detect the original or the right sequences. So there is a specific format also. So we have types of biological system which is there. We have primary database, and we have a secondary database. Now what is primary database? What is primary? From the name it suggests it's the very basic one, or the first one. So these are the primary sources of data used to store nucleic acid, protein sequences and structural information of the biological macromolecules. Some primary sources are NCBI. So our work will be basically on NCBI, basically, that is National Center for Biotechnology Information. GenBank is there, DDBJ DNA Data Bank of Japan is there, and then we have some you can say sources for the protein, like we have PDB Protein Data Bank, we have PIR Protein Information Resource, we have Swiss spots, but our main focus will be on the DNA sequences, not on the protein sequences, but our main focus will be on the DNA sequences. So primary database is basically what? These are the primary sources of data used to store nucleic acid. So on these sources, like on NCBI, on GenBank, on PDB Protein Data Bank, we have the storage, we have the abundance of nucleic acids, we have the abundance of protein sequences which are present there, and we have so much of the data which is available on these platforms. So we have to, this is the primary source of the data, and then we have to retrieve our own information on which we want to do work. So these are the primary sources of data used to restore nucleic acid, protein sequences and structural information of biological macromolecules. So this sequence collection, or the sequence collection of this database, is due to the efforts of basic research from the, you can say, from the scientists, from the academic persons who just put their data on the primary database. Getting my point? So they put their data on the primary database, and from that primary database we have to take our own, or we have to retrieve the sequences of our own point of interest. So I hope you are able to get it. Like we have, in case of DNA, we have GenBank, we have AMBL, we have DDBJ. So DDBJ is the DNA Data Bank of Japan. Then we have CIB, which is Center for Information of Biology. Then we have NIG, National Institute of Genetics. So all these are the primary source of data. Getting my point? All these are primary source of data from where we are getting the primary data, the abundant data which is there. So we have NCBI, we have NLM, which is National Library of Medicine, we have EMBL, we have EBI. So by all these methods we are able to get primary data. So let me check whether you are getting it or not. So everything is fine to you or not? Please let me know whether you are getting it or not. Yes, Shubham, good evening, Pradyumni. So just get me know whether you are getting it. Let me know, please. Everything is clear to you till now? Please let me know. Everything is clear to you till now? Please let me know. Okay. Thank you, Tamanna. Tanvi, thank you. Okay, good. Shreya, we have that much number of data because this is the basic data. So all of the scientists, all of the researchers, those who are pursuing the PhD, all will put their data on the primary databases. Getting my point? So I think the streaming screen is bit cropped. I'm not getting your point, Pratik. What you basically want to say? The streaming screen is cropped. Please check once. I'm not getting it. Is the screen is not visible to you? What you want to say? Yes, NCBI has all the information for the DNA sequences. Similarly, we have proteins. We have some another platforms in which we have the protein sequences or the data for the proteins. So I'm sorry, Pratik, I'm not getting you. You are not able to see the PPT. Unable to see the left part of the data. That is on the presentation. But this is the whole presentation. I'm sorry. Kindly refresh your YouTube maybe. Otherwise, we have the. It's the whole data which I have on the PPT. Yes. So I'm just coming back to check whether you are getting it or not getting it. But I hope you all are getting it. So that's all. Yes, yes, yes. Yes, Manish, you are very right that the primary data is the data of the researchers which feeds on the database. Basically, it is the primary data which is done by the researchers or by the scientific institutes or something like that. But it is only the thing. I have only these things there. I don't know why it is showing it to you like that. So my suggestion is to kindly refresh again. Yes, you will get PPTs Ayushi. Please, no issues regarding it. Yes, Indrani is also like that. Is also right that protein data bank from which every structure you can drive or you can retrieve. Basically, if someone is working on the 3D structure of a protein or some of these things, so you have your protein sequences as well. So okay, now let's move forward. I want to at least finish 2 topics today, so I am just doing it. And also we have okay, and also we have some small videos of 2 or 3 minutes basically, which I have to show you. But let's start it now. Okay, it may be due to screen resolution. Okay, it's an issue because of streaming resolution. Let me check. Yes, Manish, I am just proceeding. Let me small it also. Maybe because of my my picture, you are not able to get the whole screen basically. Let's proceed now. So I am just proceeding to the PPT, then I will come back and see you. OBS settings. I am also new to this OBS settings. So Pratik, if you know about it, kindly share about it along with me later. So now we will move forward further to the PPT. So yes, this is the thing that we have different types of platforms, or you can say we have NCBI, where we have the primary database, where we have the storage of the database. Like we have NCBI, we have GenBank, we have DDBG, we have PIR, we have PDB. So all these are the sources for the for the sequences, for to retrieve the sequences of DNA and protein basically. So this is that they are interconnected. They have abundance of data which is stored in these platforms. Like we have one data for data bank of Japan. They have their own data bank. Similarly, we have European Molecular Biology Laboratory, they have their own data bank. So there are lots of institutes which are working or which are storing the data. Then we have European Bioinformatics Institute also. Then, but our main focus will be on NCBI, which is National Center for Biotechnology Information. So this is the thing which is, this is the primary database. Okay, the number one. Now we have the number two, which is a secondary database. Now what basically the secondary database is? A secondary database contains additional information derived from the analysis of data available in the primary sources. So obviously secondary means it comes from the primary sources. So secondary databases are analyzed in a variety of ways and contain different information in different formats. So they have the in secondary database, basically data is analyzed in a variety of ways. We have so many, you can say, softwares are there for performing all these, for retrieving the sequences or for performing all the results. So they are analyzed in a variety of ways and contain different information in different format. Okay, so information is also present in different formats, and then we have to decode the information. So like these are the secondary databases. These are again the softwares, you can say, on which we are working. So we have primary versus secondary sequence databases. We have some reference sequence, and then these sequences, if we have some reference sequences, basically what happened, we have this is the lab, or you can say the GenBank, where there is abundance of the sequences, because this is a primary database. So we have the abundance of the sequences. Like this is the scientists, they are loading or they are just unloading lots of information here in the GenBank. Okay, from this GenBank, everyone, either the curators or everyone, they are taking it. They are retrieving the sequences of their own point of interest. Okay, so they are retrieving the sequences. The sequences which they retrieve, they become their reference sequences, that we are using those sequences as a reference sequences. We are using it for the purpose of reference only. Okay, so we are just using the data for the purpose of reference only, and then we have to analyze it either by using logarithms or by using software, like we have Unigene is there. So by all these methods we are able to compile our own results by using the method of primary and secondary databases. So this is flat file storage data formats. When GenBank, these are sources, when GenBank, AMBL or DDBG formed a collaboration, sequence database had moved to a defined flat file format with a shared feature table format and annotation standards. The flat file formats for the sequence databases are still used to access and display sequence and annotation. They are also convenient for storage of local copies. So they are just the data or the biological sequences, they are just stored in a particular file format. They are stored in a particular file format. Okay, and then it is also convenient for their storage purpose also, and it is also convenient to get the information from these particular file format. Like this is the way of information which is there in case of NCBI. Just give me, suggest me, is it comfortable if I just put off the video or this camera, because I am unable to see the laptop. So is it comfortable to you if I just put the video off, because I am not able to get to the this laptop. There's so much light on my face. Yes, thank you, because I am not feeling comfortable with this so much of light. So I am just putting it down. Yes. Just wait, 1 minute only. Yes, now I am able to see it. Okay. Yes, yes. I'm just putting unable to get the PPT basically, and there's so much light on my face also. So yes, I am here. Yes. Okay. So basically what I am saying to you, that there is a particular file format in which there is a particular file format in which we have the data format which is available. There is a particular file format, like here in this case the data from the NCBI. How the data is there, we have a header region. Okay, then we have some featureable table is there, and the last is the sequence which we have to take as a reference sequence. Getting my point? So we have the header in which we have the locus, we have the definition, we have the session number of the sequence, we have the organism from which we are taking the sequence, we have the keywords, we have the name of authors who had worked on the sequence, we have the name of title also, we have the name of journal, and even we have another important information which is there in the header region. Then we have the feature table in which we have the source, the amount of sequence, the gene on which we are working, the mRNA, their CDS code, everything is there in the tablet feature table. And last is the sequence, the retrieval sequence or the biological sequence which we have to take. So we are just going to copy these sequences, okay? When we will do it on Mega software, so we will have to take these sequences, okay? Because we have to take the data for our own data. Okay, so we have to retrieve the data basically. So we have GenBank format examples. Now this is a header region. So I am telling you about this thing. So in the header we have the locus, like the gene which they have studied. They are of 1580 base pair, and they collected it, or they are doing it on 2004. Then definition, like they worked it on Orisia sativa. Okay, they have their particular accession number, they have their source which is Orisia sativa from which they have taken the sequence, and the organism name is also Orisia sativa and it belongs to the family Poaceae. And of course there is a name of authors also right here, the name of authors also. They have the journal is there on which they publish their work, like on Science 300. In the year of 2003 they are able to publish their work, and this is the whole thing which is there in the header region. And in the case of tablet form, or in the case of table section, we have feature table identifier. We have some particular features of it which is source, gene and CDS. So we have a specific, you can say, source. Like in this diagram we are able to see the source, the organism from which we are taking it, like the organism is Homo sapiens. So the source is basically Homo sapiens. And the gene on which we are working, so the gene is like here, it's not visible. So gene is on which we are working. So like here in this we are focusing on the source, like the source is the organism which is Orisia sativa. So this is the source. And then we have a particular gene on which we are working, like they are saying the gene from 79 to 969, they are working on this gene. Okay, then we have the whole sequence which we have to take for our own analysis, or this is the sequence which we have to retrieve. Okay, so GenBank format example, sequence section. Now this is the center for all these activities, the National Center for Biotechnology Information, which is there. It is created in 1988 as a part of the National Library of Medicine at NIH. It establishes public database. So we have the data of the public basically. Research in computational biology develops software tools for sequence analysis. So, with the help of these, you can say NCBI, or with the help of all these primary databases, we are able to develop the software for sequence analysis, disseminate biomedical information. So all these are the things in case of the NCBI, or the functioning of NCBI, that they are generally established to get the public database, and they are help in the research in computational biology basically, and we are able to analyze the sequences. So NCBI database and services, we have GenBank. Then we have some another accessible sites are there, like we have PubMed, we have PubMed Central for getting the literature. To get the biomedical literature, we have PubMed, we have PubMed Central, we have Blast, we have to use it, Blast, this we have to start. Then we have VAST, which is structure similarity searches, we have softwares, and we have databases. So all these are the softwares for the bioinformatic analysis. So the GenBank, which we have to study, which is known as the Genetic Sequence Data Bank. Similarly, Japan has its own data bank, US has its own data bank, but we will do it on GenBank. So GenBank is a genetic sequence database at the National Center for Biotechnology Information, that is NCBI. It was established in the year 1982 and now maintained by the National Center for Biotechnology, which is NCBI. The DNA sequences can be submitted to GenBank using several different methods. So we have to, like the scientists, or you can say the researchers, the PhD researchers, they submit their sequences in the GenBank basically, directly in the GenBank. So it contains publicly available nucleotide sequences for more than 240,000 named organisms, obtained primarily through submission from the individual laboratories and batch submission from large-scale sequencing projects. So everyone is, or each one is working in the lab, or has is pursuing his PhD or something. Then they are able to they are able to submit the sequences in the GenBank. Okay, are you able to get it? I'm sorry for the intermediate break. So is Pallavi is here? Pallavi, are you here? Pallavi, Pallavi, are you here? Pallavi, are you here? If you are here, please reply. You are there in the WhatsApp group. You are putting the messages on the WhatsApp, but right here I am along with you. So please let me know if you are here. Then please share what you want to ask. I'm sorry to interrupt in between the class, but you are putting the message on the WhatsApp group. So are you here? Please let me know. Pallavi. Pallavi or Megha or Pratiti, Stuti. So are you here? You are putting the messages, Pratik, Tavleen, Valina. So you all are here. So please share me what's your problem, what you basically want Tanvi or Pradhumi. Please share it with me also. You are putting it on the WhatsApp, then who will reply you on the WhatsApp? I am here. So please, if you are able to share your point of view with me, then I will definitely help you. Please let me know what your issue is. So you all are putting the messages on the group. Like Valena is there. So all of you are there. Even the students have moved out from this online class and they are going to the WhatsApp group. So any one of you is there, please let me know. Are you able to listen to my voice, Indrani? Are you here? Indrani, are you here? Are you able to listen? If you are able to listen, then please reply. They just moved from the online class and they are just they are just typing on the WhatsApp group. Pratik, are you here? Pratik, please let me share your query or if you have some issue because you all are putting it on the group. Just share your problem here along with me, please. They are saying it's very disappointing. Yes, yes, dear. Please share your. Yes, yes. Please share point of view. No issues at all, dear. I am here for all of you, so you should not be worried. Even if I have to take 10 to 15 days, I will take it, but I will cover each and everything. So you should not be worried. Getting my point? So you should not be worried about all these things. I will cover it. But why? Yes, I want to cover. I will want to discuss it after the class. But they are just typing on the group. If you are Prasthita, if you are here, just open the WhatsApp group. They are typing and typing in between the class. They are just typing in between the class. So Pallavi Dhal, if you are here, please reply to me. Yes, Priyashri, I am just replying to you. Either it will take more than 10 days, it will take 10 to 15 days. I will definitely cover it. So please don't worry, dear. Please. Yes, Pradyumni, what is your issue? Please let me know, Pradyumni. I am your teacher, so you should be comfortable in letting me the problem, please. Okay. Pile is saying most of us have covered these basic topics and we signed up for the course primarily because of other advanced courses such as drug discovery and proteomics. Yes. Yes, it's my request to all of you, Pallavi and even the other one. Please don't put it in between the class. Just even if you know it, then just listen it once again, and I will definitely listen to your issue after the class because now we have doing something and we are doing it with full concentration. So it's my request, do not put any messages on the group because by this we are able to discuss everyone. Okay, so if you have any issue we will discuss it. Please don't worry, please. So if you have any issue, like we have Prakriti, we have Rohan Kotak, Pallavi Dhan, Valina, are you here? Savleen, Praditi, and I will listen to all of you. I will listen to all of you. So please don't worry. I will listen to all of you. Just give me some time. So please don't worry. I will. Okay. So we will discuss everything. Just wait for some time. And if you are listening, then please concentrate on the topic. After the class, we will discuss everything. Okay. So please don't be panic. I know you are expecting something else, but we will definitely cover everything. So please don't be panic. And please don't put any messages on the group. I will reply you here and I will help you. Okay, that's for sure. I will help you. So please don't be panic. I will help you. If you have any query, then you will tell me later on. Okay, so let me complete the PPT. Please don't spoil it in between because we are doing it with full concentration. So I am just on this GenBank. I am telling you about NCBI databases and the services. I am telling you about the primary sequence database which is there, and we have GenBank which is genetic sequence data bank. So GenBank is the genetic sequence database at the National Center for Biotechnology Information. It was established in the year 1982 and now maintained by the National Center for Biotechnology, which is basically NCBI. So DNA sequences can be submitted to GenBank using several different methods. It contains publicly available nucleotide sequences for more than 240,000 named organisms obtained primarily through submissions from individuals, laboratories, and batch submissions from large-scale sequencing projects. So they are getting the data from the individual researchers, even from the the project work which is there, or from the large lab basically. So it has a flat file structure, that is an text file readable and downloaded by both humans and computers. There are two main ways of making batch sequence submissions to GenBank. We we have NCBI barcode submission too, which is there in the NCBI. I will show you when I will open the NCBI, and we have another tool which is Sequence. So by both these tools, we are able to upload our sequences. Getting my point? Then we have, like, this is the thing. So we have the resources, like if we want to do it on, DNA or RNA, if we want to work on DNA and RNA, then we have the GenBank. So in the NCBI, you will go on the resources button, and then you will get the you can say, you have to click on DNA and RNA, and you have these softwares for work it on the RNA and DNA sequences. So this is the GenBank basically, which is there for the nucleotide sequences for DNA and RNA basically. So you have to put your sequence here, and then you have to do the search. Like you want to retrieve the data related to humans, so you have to type here Homo sapiens. Okay. So if you want to retrieve the data related to dogs, then you have to put the name of any, or even if any plant or any medicinal herb, anything is like that. So you have to put the scientific name here, and then you have to do the search. Okay. So this is our GenBank, or you can say NCBI. In the NCBI, you have to go to the resources. Within the resources, just go to the DNA and click on the DNA, and then you are able to get the GenBank. And on the GenBank, you are able to get the, or you are able to retrieve the sequences. Then we have EMBL, which is known as European Molecular Biology Laboratory. It is a molecular biology research institution supported by 22 member states, 4 prospect and 2 associate member states. So this is the similar, same like GenBank, but this is the European Molecular Biology Laboratory. So they have their own data stores. So they have their own, in which they are able to store their own data. Okay. So AMBL was created in 1974 and is an intergovernmental organization funded by public research money from its member states. So, EMBL is basically what? This is the site of the EMBL nucleotide sequence database. You have to click it, and then you are able to access the site. It is used to incorporate and distribute nucleotide sequences from public sources. The database is a part of international collaboration with DDBJ, which is Japan, and GenBank, which is the software of USA. So data are exchanged between the collaborating databases on a daily basis. The web-based tool, which is Webin, is the preferred system for individual submission of the nucleotide sequences, including third-party annotation or alignment data. Then we have then we have I think some of you are not able to see it properly, so I will put it like that only, because some of you are not able to see it properly. So It is used to incorporate and distribute nucleotide sequences from the public sources. The database is a part of an international collaboration with Japan and USA. The data are exchanged between collaborating databases on a daily basis. The web-based tool Webin, so we have a tool which is known as Webin, is the preferred system for individual summation of nucleotide sequences, including third-party annotation and alignment data. So we have this software which is EMBL, which is European Molecular Biology Laboratory. So you have to click it on it, and then what you will get, you will able to open the software which is EMBL. And in this software, these are locations of EMBL, like their softwares, or they are basically EMBL. They are located in Germany, they are located in France, and also in Italy and UK. So what is there in EMBL? Yes, this is this. So when we click on the EMBL on Google, we will able to get this page. On this page you have to find a gene, protein, or a chemical. So you have to write the name of a particular gene or a protein, of a particular species or organism on which you want to work, and then you will have to enter, or you can say you have to do the search, and then you will be able to get the results also. So it's my request to all of you that after the closing of this session, just go through all these softwares like GenBank. So you have to go on to the Google and you have to type here. Uh, so you have to, like, on the Google you have to type it like NCBI, and similarly you have to type EMBL. So by typing on NCBI, and then you have to go to resources. So just by this you have to go there, and if you are able to go in the NCBI, which is National Center for Biological Information, this is taking the time. Let me get what you are saying. Tavleen, I will discuss everything with you, dear. Please wait for some time, please. Yes, yes, I know. Please don't worry. Please don't worry, please. I will discuss everything. I know what you want to say. So this is the NCBI, and we have all types of databases here. We have on the targets which we want to do, and here we have to search the nucleotide bases basically, or even you can do it by using all databases also. And here we have all the databases on which we want to do. And you can type here. Yes. So we have the resources which is DNA and RNA, like we want to do it on in case of, or we are interested in DNA and RNA sequences. So just we have to go there, and then we are able to type our own data, or our own, you can say our own organism, or the sequences in which we want to work, then we will get the result. So this is the European Bioinformatics Institute. We have to go there, and it's my recommendation to all of you, just kindly go on these sites, and just search something, anything you want to search, just go there and search. Then we have, this is NCBI Blast. This is another thing which we have to discuss thoroughly. We have to do blast and FASTA as well. Many of you have done it before also, so that is why they are feeling a little bit sad. Then we have another site which is PubMed. So what basically PubMed is? PubMed is a free search engine accessing primarily the Medline database of references and abstracts on sciences and biomedical topics. The PubMed system was offered free to the public in 1997. The United States National Library of Medicine The National Institute of Health maintains the part of the Entrance System of Information Retrieval. PMID is a unique identifier number which is used in. They are assigned to each article recorded when it enters the PubMed system. So when you are going to the PubMed, you will able to get each article which have their own reference number which is given to it. The PMID is always found at the end of the PubMed citation. So this is the PubMed, or you go to the resources and then go to the literature. Like in first we go to the DNA and RNA, but now we have to go to the literature and we will go to PubMed. Then this PubMed will open, and from this PubMed you are able to get the literature or any type of you can say help. Like they are just typing immunoglobulin structures and then just type the search, and they will get 807618 results. Okay, so that much information is present, or that much literature, you can say the literature basically. So that much literature is basically present on the PubMed. Okay, getting my point? So that much literature is present on the PubMed, so you can type anything. Even if you want to type the title of your review paper, then it's also okay. So you can type the title of your review paper, and then you will get lots of important paper here. So it's my suggestion to all of you to kindly go through all these online sites and just use them, just try to use them. Okay, so we have to type it, like if you are working on CRISPR or something, so you have to just go to the search and you will get the result. So these all are the papers, and you will just have to open it and you will get the good content. Like this is the impact of donor specific anti-HLA antibodies and antibody mediated rejection on outcome after intestinal transplantation. So we have the proper abstract here, and even we are able to download the PDF, not in the all, but some are freely available, so we are able to download the PDF also. So you have to go in the NCBI. In the NCBI you have to go to the resources, and in the resources you have to go to the literature and then PubMed, and then here you have to type your own article. Like we are doing in PubMed also, we are able to do it. So you have to type the title of your article, and then you have to search the paper related to your own. So this is the thing, like clinical feature of neuromuscular disorders in patients with N-type voltage gated calcium channel antibody. So it's my suggestion to all of you to please kindly go through PubMed for literature searching, or you can say for literature searching basically. So this is PubMed. Similarly, we have European Journal of Translation Biology. So from these also we are able to get the, like from here we are able to download the PDF and we are able to get the results. So you have to go to NCBI, and in the NCBI you have to go to GenBank number 1, and in the NCBI then you have to go to the literature and then to the PubMed. So these are the softwares which you have to go through by your own self. Even we have one another which is European, which is this one, EMBL. So you have also typed this European Molecular Biology Laboratory. So you have to type it on Google, and then you will have to type some specific name of organism here, like here, or a gene or a protein in which you want to work, and then you have to click the search, and then you are able to get the sequences. So I think you are able to understand the thing which I want to say. So you have to go to all these online sites on PubMed, GenBank, NCBI, and also the European one, EMBL. So these are databases which is covered by. So we have GenBank, we have reference sequences, we have PDB, we have lots of platforms are there in which data sequences are saved, or in which lots of number of sequences are available. Like if you want to do taxonomy, if you want to work on taxonomy, then we have NCBI taxonomy in which you will able to get your data. If you want to search the literature, we have PubMed. Okay, if you want to do the 3D structure of the proteins, then we have MMDB. So all these are the software which are available on the internet. So you will just type your own query and you will get the result. Like search here, they are putting it immunoglobulin and they will do the search and they will get the result. Like by typing immunoglobulin, it is basically a protein. By typing it, they are getting the results of 580708 within, I think within 1 second or within 2 seconds, we are getting that much of result. So it is my request to all of you to go through all these softwares, okay? Then if we are clicking it further, if we are clicking the number one, that means this one, if we are clicking it further, then we are able to get the complete information about that particular gene or a molecule. And even on the PubMed, we are able to get the literature of this one. Third, three-dimensional structure of a human immunoglobulin with a hinge deletion. So, three-dimensional structure of a human immunoglobulin with a hinge deletion. So, we are able to get the literature of it also. That means we are able to get the paper of it also. If the author has done the paper on it, then we are able to get the paper. So, like this is, they have the paper which is on the sequences which we are retrieving. So, you are from here you are able to get the, you can say you are able to search the literature also. Then we have some SRS, which is also the sequence retrieval system, and retrieval system developed by EBI. It is also a software. More than 250 data banks have been indexed, more than 35 SRS servers over the WW or worldwide. So this is again platform where we are able to get the sequence and the protein. We are able to search the sequence related events, or even we are able to get the bibliographic results. This bibliographic is also in your syllabus, so I will cover it also. So this is the SRS, and on SRS you are able to get many another softwares, and like cluster is there, FASTA is there, blast is there. They all are interrelated. Similarly, we have PDB software basically for the proteins, for the protein data. It is a protein data bank for the protein structures, and they have their own working. So are you able to get my point or not? Please let me know. Yes, Tamanna, you have to just type it on the Google NCBI, like I discussed it before also. So you have to type NCBI, and by typing it, you will get the result. And then if you want to, like there is a PubMed, if you want to search anything, okay, if you want to, I have some little bit problem in my connection basically. So if you want to, uh, let me check whether it's opening or not. No, it's not opening. So we have NCBI, we have all databases, then we have PubMed for literature search. So just go on the PubMed and just type your name of your own article or your own topic. So I'm sorry it's not opening right now. So just you have to type here, like you want to do it on Homo sapiens. So you have to type Homo sapiens, which is human, and Homo sapiens, like gene, the name of a gene, the particular gene on which you want to do work, or on the particular gene or the sequence which you want. Then you have to do search here. I have some connection problem, that is why I am not getting it fast. So you will have to do it on NCBI. In the NCBI we have taxonomy also, if someone is working on a taxonomy. Otherwise we have the literature, and in the literature we have PubMed. Okay, so you have to go to all these sites, and then you have to tell me tomorrow whether you are able to get something or not, whether you are able to get the sequences or not, because right now I don't tell you anything about the sequences. So it is your tomorrow task, that tomorrow when we will meet, you have to show me your sequences. Okay, any sequences, whether it is related to humans or something, I think it will take some time. Let it open. So tomorrow when we will meet, we have to do, you definitely have some sequences. So I just want to show you one PPT which is there for this bioinformatics, which will, by which you are able to get it more about the bioinformatics.
What is a database? A database is a collection of information that is organized so that it can be easily accessed, managed and updated. Databases are composed of computer hardware and software for data management. The chief objective of the development of a database is to organize data in a set of structured records to enable easy retrieval of information. Although data retrieval is the main purpose of all databases, biological databases often have a higher level of requirement, known as knowledge discovery, which refers to the identification of connections between pieces of information that were not known when the information was first entered. For example, databases containing raw sequence information can perform extra computational tasks to identify sequence homology or conserved motifs. These features facilitate the discovery of new biological insights from raw data. According to a report of 2014 Molecular Biology Database Collection in the journal Nucleic Acids Research, there are a sum of 1,552 databases that are publicly accessible online. This does not include online services without publication in peer-reviewed journal or being developed by commercial companies. Thus, the actual number of online databases could well exceed this number. Considering the continuously proliferating number of biological databases, it becomes increasingly daunting and time-consuming to navigate in the huge volume of databases of interest. Considering the heterogeneity in data type, scope and curation, biological databases can be classified into multiple categories under different criteria. According to the scope of data coverage, biological databases can be classified as comprehensive and specialized databases. Comprehensive databases cover different types of data from numerous species, and typical examples are GenBank, European Molecular Biology Laboratory, EMBL, and DNA Databank of Japan, DDBJ. These three databases were established as the International Nucleotide Sequence Database Collaboration in 1988 to collect and disseminate DNA and RNA sequences. On the other hand, specialized databases contain specific types of data or data from specific organisms. For example, Wormbase is for nematode biology and genomics, and RiceWiki is for community curation of rice genes. According to the level of data curation, biological databases can roughly fall into primary and secondary or derivative databases. Primary databases contain raw data as archival repositories such as the NCBI Sequence Read Archive, SRA, whereas secondary or derivative databases contain curated information as added value, for example, NCBI RefSec. As a consequence of the explosive growth of data, curation increasingly requires collective intelligence for collaborative data integration and annotation. Therefore, biological databases can also be classified as expert-curated databases, for example, RefSec-Enter, and community-curated databases, which are curated in a collective and collaborative manner by a number of researchers, for example, kernelwiki and GeneWiki. Finally, according to the types of data managed in different databases, biological databases can roughly fall into the following categories: DNA, RNA, protein, expression, pathway, disease, nomenclature, literature, and standard and ontology. Despite having highly different functions, almost all biological databases share similar architectural. Each consists of 3 tiers of software. At the bottom is a database management system, DBMS, that manages a collection of facts. At the top is the web browser that transmits requests for data to the database and renders the responses as web pages. In the middle is a software layer that mediates between the DBMS and the web browser to turn data requests into database queries, and to transform the query responses into hypertext markup language. HTML. In addition to the gene database and PDB, there are many more biological databases designed for different purposes. The table here shows some of the most commonly used databases in bioinformatics that might help you in your investigation. However, one of the major problems is that each of these database resources contains a different subset of biological knowledge. Although each database can answer questions in its domain, it cannot help with questions that span domain boundaries. For example, the Fly-based database does not know about orthology relationships. The Institute for Genomic Research, TIGR, database can help with orthology but not with map position, and the UCSC database knows nothing about the fly. As this example shows, researchers must become adept at database surfing to answer many reasonable questions. Imagine, life would be much simpler if there was a single biological database. But this would be a poor solution. The diverse databases reflect the expertise and interests of the groups that maintain them. A single database would reflect a series of compromises that would ultimately impoverish the information resources that are available to the scientific community. A better solution would be to maintain the scientific and political independence of the databases, but allow the information that they contain to be easily integrated to enable cross-database queries. There are many integration challenges. One of the most difficult is the one that might seem the most minor. How do you assign and maintain the correct names of biological objects across databases? For example, consider the DNA damage checkpoint pathway gene that is named RAD24 in Saccharomyces cerevisiae or budding yeast. Saccharomyces pombe or fission yeast also has a gene named RAD24 that is involved in the checkpoint pathway. But, it is not the ortholog of the S. cerevisiae RAD24. Instead, the correct S. pombe ortholog is RAD17, which is not to be confused with the similarly named RAD17 gene in S. cerevisiae. Meanwhile, the human checkpoint pathway genes are sometimes named after the S. cerevisiae orthologs, sometimes after the S. pombe orthologs, and sometimes have independently derived names. A more subtle problem is the clash of concepts as users move from one database to another. For example, the use of the term pseudogene, genomic variant, and even the concept of the gene itself can mean radically different things to different research communities. Some researchers treat the gene as the transcriptional unit itself, whereas others extend this definition to include up and downstream regulatory elements, and still others use the classical definitions of Cheestern and genetic complementation. There are also technical challenges. The various biological databases use different DBMS and none provide a standard way of accessing the data. Even more challenging is the issue of updates. Biological databases are always changing, so integration must be an ongoing task. There are main ways in integration of biological databases can be achieved. First, link integration. Link integration has been by far the most successful, because it lends itself to the haphazard nature of the web. In the familiar and ubiquitous version of this technique, researchers begin their query with one data source, and then follow hypertext links to related information in other data sources. Second, a variation on link integration is embodied in the popular sequence retrieval system, which is a keyword indexing and search system for biological databases. Sequence retrieval system is more sophisticated than general web-based search tools, for example, Google, because it recognizes the existence of structured fields in source databases and allows maintainers to explicitly relate a field in one database to a differently named field in another. Unfortunately, link integration is problematic on several fronts. First, it is extraordinarily vulnerable to naming clashes and ambiguities. For example, a naive researcher who tries to navigate through the RAD24 gene confusion using SRS or direct database to database linking might wander into the wrong gene family. Second, there are update issues. An outgoing web link represents a leap of faith that the page at the other end of the link is still valid, or if it
So it is a quite big PPT, big videos. I'm just sharing the I'm just sharing the link. So you have to watch it by your own self. Okay. So when you will be free, so you are able to watch it again. Okay. Yes. Yes, these databases are free of charge. I'm just sharing the links with all of you because it takes lot of time. But these videos are quite important for all of you. So you have to go through with all these
It can be
basic
of
videos.
16
Okay.
is
So I'm just sharing it with you. So I hope you will be able to get it. I hope you will be able to see it by your own self. Because these are important ones. By this typing on the Google or on the NCBI, you are able to get the results. Even if you want to do the PubMed, you have to go to the literature, and in the literature you have to go to the PubMed, and in the PubMed I have my signals are not good here, and on PubMed you will able to search even your own review papers, the review papers on which you all are working, so you are able to search your own review papers. Getting my point? Or even you are able to get the literature or the good content for your own review papers. So that is my request to all of you. Just kindly go through all those these databases and just find your good review papers there. Okay, so I am not going to show you the videos because you will be able to see it by your own self. Getting my point? Now, yes, now it's 9:30. Now you will be able to tell me what's your point of view. You want to do on drug designing also, because the syllabus which is given to me or the syllabus which I have is totally different with the syllabus which you all have. So if someone of you which is there in the group, if you are listening to me now, please let me know what you basically want to say. Okay, because we have another term which I will start tomorrow. The same things are there, but a little bit more advanced. So what is your problem? Like this is again on the PubMed, they are writing the name and they are getting the literature. We have the primary sequences there and we have the secondary sequences also there. So from here there is sequence file formats. I have to start my next class which is on FASTA, and also we have BLASTA on this time. So now if, yes Tavleen, you are here now, let me please share your query. You want to do on drugs basically. Please let me know Tavleen, and I think some more students are also there. So please be comfortable to share your point of view with me. What basically you want, please let me know. Then I will help you definitely. If I will come to know about the queries, then I will try my best to help all of you. Please let me know. Tavleen, even one more is there. Pallavi, are you here? Please reply. Here, dear, I just all. Don't be panic or don't feel angry on me that I am not replying you, because this topic, this sequence alignment, we have to discuss it on tomorrow. That is why I am not replying you. I will show you like this FASTA. This is the format which we have to do. Okay, after it we have to do the alignments. Okay, so don't worry dear, I will cover everything, please, because this mismatching part is not coming today, so that is why I have not discussed it today. We have to do it tomorrow. Like this is alignment. Okay, we have some mismatches there, like in case of G we have S here, if you are able to see. So we have some mismatches is also there, and I have to take it tomorrow. So please, we will do the examples also, so please don't worry, we will do these alignments. Okay, but don't be angry on me, please don't be angry. Then we will discuss about these alignments. Then I will answer your query. Please wait for tomorrow. Okay. She is saying for the fifth time, ma'am. Why in scoring a sequence alignment mismatch is 0 and gaps are -1? I will definitely answer there tomorrow, Ria. Just be patient because we are not taking it today. We are not doing the alignment today. That is why I am not taking your question. Please don't mind. Please don't mind. Okay. So I will answer it tomorrow when we will do the pairwise alignment and when we are doing the another forms of alignment, local alignment, global alignment, then I will reply there. Please don't be angry on me, please. I will definitely reply. Okay. Yes. Yeah, I understand, Taween. I definitely, I am with you. I am with you. Even I checked the syllabus which is there on the WhatsApp group, which is there. So yes, it is totally different from the the syllabus which I have along with me. So I think this thing should not be there, and I will talk with the sir also that please do something. They have some topics like drug designing. They have some important topics basically. I have not go through all the topics. Yes, they have some important topics because you all are aware of bioinformatics. You must have done it before also. So I will request them. Yes, you have protein structure prediction, proteomics, introduction to drug discovery, cell cycle, key to drug discovery, structural biology, drug design, drug targets. Yes, the topics are very advanced. Yes. The topics are very advanced. But don't worry, I will convey your message. Don't be panic. I will even request from my side also to the team that please cover all those topics. Okay. So please don't be panic. Hmm. Yes, Shruti Singh, ma'am, the syllabus which we paid for is completely different from the topics. Yes, Shruti, it's completely different. I agree with all of you. They are completely different topics. Like they are very advanced topics. We are discussing very basics. We are discussing about the FASTA, about the BLAST, about NCBI, and GenBank. Even those topics are new to some students which have not done it before. So those topics are new for those students also. So that is why I have to start the very basic things because there are some new students also. But the topics which are there in the syllabus, they are quite advanced topics like protein structure prediction, RNA structure prediction. They are quite advanced topic, and I agree with it. I agree with you on this thing that yes, you are right. No issues, Praveen. Payal, I will. After this session, I will even call him, and I will let you know that please, they have some, they have different things they want to learn. So I please don't panic. No worries. Please. Yes, Manish, we will discuss. No issues. I will discuss everything, and we will do it practically also by doing it on the Google. Okay, no issues at all. But of course, for tomorrow, you have to search about NCBI. The GenBank is there, and then you have the PubMed. So all of you have at least five references from the PubMed. Okay, so you have to type your topic on the PubMed, and you have to save it. Okay. No issues, Kruti. I will after this class, I will call them so that the issue should be resolved as early as possible because it is creating the problem for all of us. Even some students are new here, so we are doing something or we are concentrating or focusing on something, so it is creating the problem for all of us. I will try my best. Okay, the same thing happened with proteomics also. Okay, they change the group settings. Okay, yeah. So you are also there for the proteomics session also. So you are also Yes, Arif, we will cover the local and the global alignment also. Manish is saying Edufabrica is not professional. No, no, dear, it's not like that. Hi. Sometimes what happens, miscommunication is there. Maybe this is the only problem, so please don't worry. Everything will be resolved, and it should be resolved as early as possible. Yes, training project is so confusing. I also don't know about this, Ankita, because there is one more term which is internship is there. So only they know. I am not. I am just a trainer and I have to convey. I have to deliver the lecture. That this is the thing which I only know. Ma'am, can you change the class time so that we do not feel done at the beginning of the class? Oh, Madhumita, what do you want to say, dear? That means we have done very little today. This is the thing which you want to say. Yes, yes, Tavleen. Yes, definitely. I will call them and I will, I will put your point that you already done the basics of bioinformatics and now you want to do the advanced course basically. Okay, Manish has some answer that training is the theory part and project is the review paper writing and publication. Yes, yes, this is the thing that training is this 20 hours class and then we have 10 hours for your review paper which is come under the project. Hmm. Manish is saying YouTube is consuming a lot of my data and is buffering most of the time. Yes, this is the disadvantage of basically the online platform. We are not able to see each other. This is the problem. Yes, Valena, I'm not angry with all of you. I know you are my student and you all are dear to me and you also take care of me also and even give me best wishes when I am ill. And you have the right to say if something is wrong. There is no issues at all, dear. I know you have some experiences, so this is your right to share your experiences. No issues at all. Yes, yes. They are not even replying, and they are removing the person from the group. Yes, yes. I am also agree with Valina. The main thing is basically what that you all have done the basic part. So it is very basic to you that we already done it, and now you have joined this session for the advanced part, which is a little bit advanced, but the thing which is the as a teacher I have my own topics which I have to cover and even there are some students which are new, so I have to start it from very basics. And even for you also I am concerning that how we will cover this drug research part or something like that. So I will have to discuss it seriously because on the very first day I don't think this become that much complicated. Yes, yes, I will have to resolve it. Otherwise, it is just ruining the whole class. You are not feeling comfortable, and then I am not feeling comfortable, then the focus gets disturbed. Okay, okay, and also Pyle is saying agree with Manish on data consumption. Yes, yes, even on this, on this timing from 8 to 10. So we are here for all of you, for helping all of you. So please, don't be impatient. I will discuss everything with sir and with ma'am also, and I just want to solve it. Yes, I want to. I don't want that my students become panic. I don't want this. Yes, this is my first and the foremost duty. Yes, so no issues at all. Even if I am unable to help you regarding the topics, then I will ask them to arrange someone so that your topics will be clear. I don't want the loss of any of the students. You all are very good students, and you all are here at 8 p.m. sharply. So definitely you will get something. You have to get something. Mm. Chesney saying, "Ma'am, is the class over?" Yes, dear, it is over now. It is 9:40 now. Plus, please don't be, you know, feel uncomfortable that we have done little part. I have my presentation and we have 181 slides and we are on 164. But in between, some students have some queries, so that is why I have to stop in between. So please, please, please don't be panic. I am here for helping you all. So please, I will just talk with them and then I will let you know, or even they will reply you. You will not have to take any type of worry. Okay? So please, please don't take any type of worry or tension. So is there any volume or not? Are you getting my voice or not getting it? No, no, Palvi, no, please don't say like this that you have no faith. Yes, sometimes what happens is miscommunication basically. So please don't be sad. I don't want all of you to be sad. Sometimes what happens, there's a miscommunication, there's a gap in communication. Okay, so don't worry, I will try my best. Please don't be sad, or I know you all have what I say now. So please don't be sad, and let me try this. Let me ask them and then I will let you. I have your syllabus. I will discuss everything and then I will let you. Okay. At that time, on the meanwhile, that we have on these things, when we are not able to concentrate on the topic or when we are not able to get anything, at that time you should be focused on your review paper. Okay, so if you are not feeling well enough or good enough for the topic, so at least just focus on your review papers, please. Okay, if you are not comfortable in the class or in the session or on the topics, no worries, no issues, don't be panic, don't be sad. Utilize the time on writing the review paper. Getting my point? Instead of discussing the things, just write the review paper. So utilize all of your energy. If you are uncomfortable in getting with the sessions, uncomfortable with the topics because they are basic, so rather than focusing on it, just focus on your review paper. So please select your topics and just focus on your review papers. If you are still uncomfortable regarding anything, XY, anything, you have to write, you have to complete your review paper. Okay. So just complete your review paper. Pallavi and Tavleen and Vayona, all of you all are my students. So I am just suggesting you that please start writing the review paper. Okay, if this part is bioinformatic part is not suitable for you, at least just write the review paper. Even Indrani is here, she also done the basics, or she also done the bioinformatics, lots of the parts of bioinformatics, but still she is listening. So thank you Indrani for your support. So I think that's all for today. Let me just clear the issue first. Okay, so please don't be panic. I am there, and I will try my best to help all of you. So even for the next session or the next classes, I will be there or not. But I want that there should not be any loss of all of you. Okay. No, no, Tabir, no, no. Please, don't focus on that. They just block the messages or they are not replying. I am saying utilize your energy in the form of review paper. Okay. So you should not to think about the negative things. Rather, utilize all of the energy and put it on the review paper. Getting my point? So just, and this is my request to all of you. Don't feel disappointing or don't feel bad. You just put your point. That is very good thing, very good thing. Now just focus on the review paper, okay? Because this review paper will be your 2020 event or your 2020, the thing which you have done in the year 2020. So please, yes, Manish, I know. Yes, it causes a problem when we are only discussing the issues. Manish, just support or be with me and trust me. We will definitely get everything best, and we will do something, and not something. We will do everything good. So yes, I will try my best to make 2 hours interesting and knowledgeable. Just give me some time because some students have some problems, and as they are my students, so I have to handle those problems also. Please try to understand. We will from tomorrow onwards we will do it fully. Okay, so please don't be sad, and it is my request to all of you that don't be sad. Instead of focusing on the negative things, we have to put those energy into the positive way. So we have to put it in the review. So it will become your achievement in the year of 2020 that you have your one research paper or your one review paper in your hands. So just focus on the positive things. Okay. Yes, Indrani, you can message me on the WhatsApp also, no issues. All of you can message, no issues at all. Yes, you all will, yes, dear, you will be added again, no issues at all. You will get everything, the materials and notification, the link, everything is provided to you, no issues at all. Just be patient. I will let me call them first, and then I will reply you. Okay? So please concentrate all of your energy in a positive way and put it in a review. Okay? Regarding your topics, I will. Yes. Praduma is happy, and by seeing their smiley, I am also feeling happy now. So please be happy. Please don't be sad. I will take forward your queries, your questions, but please don't be sad. Okay. Rather utilize whole of your energy in the review paper. So that is the today's discussion or few words from my side, and those who are new ones, they have to search on NCBI, that is NCBI, and then GenBank is there, PubMed is there, and all of you should search some topics related to your own topic and show it to me tomorrow. Okay, so that's all for today, and no issues. We will meet tomorrow with lots of fresh energy and fresh vibes, and take care of yourself. Okay, Indrani, just take care of yourself as you have to go. You had to travel to from Kolkata to Chennai and then to Pondicherry. Oh my God, it's a long traveling. So take care of yourself also, and let me know when you will reach your home or in Pondicherry. So please let me know also. So that is all from my side. We will meet tomorrow with lots of fresh energy and fresh vibes and lots of new things to do. So take care. Bye bye. Good night. Okay. Please don't be sad. Please. So take care. Bye bye and good night. All of you. Okay, dear. Okay, Indrani. Okay. So no one will now become panic or no one will do anything. All of you will have to write the review only. Okay, so we will not discuss anything regarding the topic. I will discuss it. So you have to write the review paper only. Okay. So that is all from my side. Take care of yourself. Bye bye. Good night. So good night to all of you. Good night. Please take care of yourself. Don't be sad now. Okay. Don't be sad. I will talk. No issues. Please. Those who are feeling sad, please don't be sad. Please don't be sad. I am there along with you. So just if you are now free, as it is 9:49 only, you are free now. So just write your review paper. Please just focus on your review paper, dear, all of you. So that's all from my side. Okay, bye bye. Good day or good night. Take care of yourself. Bye. Okay, Indrani will reach today evening. Okay. She reached today evening. Okay. So good night to all of you. Good night. Bye bye. Take care. And also subscribe to my channel. You are not subscribing to my channel. Then how you will be able to get the notification? So please subscribe to the channel with crash courses. Please, all of you. Nasir is saying yes, ma'am. Good night. Okay. Good night. Good night. Good night to all. Good night. Take care. We will meet tomorrow. Okay. Bye bye.